Good morning to everybody. Myself, I am Dr. G.P. Bhavani, Head Department of Chemistry, respected guest, Dr. M.KARTHIKEYAN, respected Secretary, Lalitkumar O.J., respected management, respected Principal, Dr. N. Sujata, dear colleagues, participants, and my dear friends, a very good morning to one and all present here. It's given me a great pleasure to welcome you all. Our institution, Sri Chandraprabhu Jain College, conducted many more programs like online and offline. Main motivation, exploring the knowledge of the students in and around the world. Not only the knowledge of our pupils are easily competitive in a successful world to achieve something. First of all, I wish to appreciate to have interest in exploring the knowledge in a session. By joining our online sessions organized by WeCan Institute, the international seminar, 1 week virtual mode, emerging trend in chemistry. This is the last day international seminar. I thank everyone who actively participated last 1 week. May I think it's a very useful all of us. I welcome our special guest, Dr. M.KARTHIKEYAN, Associate Professor, Pharmacogenomics and CADD Laboratory, Department of Bioinformatics, Alagappa University, Karaikudi, Tamil Nadu. They give you a topic of presentations, emerging trend in chemistry in a bioinformatics via and talking process. It's a wonderful, very useful topic in silico study in our day-to-day life. Thank you, sir. Once again, all the participants, hearty welcome. I hope all of you enjoy the sessions. I briefly introduce our chief guest, Dr. Karthikeyan Uttuswamy, sir, Associate Professors, Pharmacogenomics and CADD Laboratory, Department of Bioinformatics, Karaikudi, Tamil Nadu. He has completed and qualified PhD in biomedical genetics from the Department of Genetics, University of Madras, Taramani campus, Chennai, India. He has completed MSc in biomedical genetics, the same university, University of Madras, Taramani campus, Chennai. He has a professional experience in teachings more than 18 years, in a research 24 years. He has a research supervisor. He completed more than 7 number of PhD and ongoing of 5 numbers. He has a completed MPhil students project 8 numbers and MSc in 45 numbers. He had published 119 numbers of publications, journals, and national level of 8 numbers. A research matrix is a citation of 1545, H index 22, and 110 index of 552. Cumulative impact factor 239.217. He completed the major project. He got 309 lakhs rupees. An ongoing project, 360 lakhs he proceeded. He had published more than 6 books and 4 chapters. He has attended more than 40 conferences, national and international, 81. He had got more than 16 awards from the various deputed field. He has other contributions. He has a deputy coordinators, life members of treasurers in a bioinformatics, drug discovery society, membership in more field, membership in more institution of biosafety committees, associate and assistant directors from the international and Alagappa University, deputy coordinator and different field chairman, members and chairperson of different field. Thank you, sir. He recently published more than 18 papers. Thank you, sir. I hand it over the session to our respected principal, madam. Ma'am, please proceed, ma'am.
Thank you, ma'am. A warm welcome once again on behalf of the management and myself, Principal Dr. M. Today, the last session of the international seminar, which has been organized by the Department of Chemistry. So, my wholehearted congratulations to the department convener and the head of the department, Dr. G.P. Bhavani, for having organized on the new concept of learning in chemistry. I hope all the students, those who are participated in all the session which has been conducted throughout this week, have taken a good insight from the various eminent speakers of the various fields. My profound thanks to all the people given their valuable time and present their session in this international seminar. Dr. J. Sai Chandra, Dr. Vaidyanathan, Dr. Mahesan, Dr. Isaac Devakumar, Mr. S. Tulasidasan. Today's session by Dr. Karthikeyan. We are very much and pleased to thankful to us, thankful to you, sir, for having accepted our invitation and presenting today's session. So thank you very much, all the personalities who are joined in this international webinar organized by the chemistry department. So dear students, so take the very enriched knowledge by the various speakers, and so you have to apply in your life as well as in your so you have to learn the life skills through this people. Thank you all very much. I am happy to be the part of this program. Thank you.
Thank you, ma'am. I invite our guest, special guest, M. Karthikeyan, sir, do the special topic presentation. Sir, please proceed,
yes,
sir.
Yes, ma'am. Thank you, ma'am. Thank you for the opportunity given to me. And also today talk, it is on computational chemistry. How we are using the chemistry part, chemistry in the different field of our drug discovery. What we are doing is a drug discovery is a prime important of this. Thank you for the first opportunity. Today I'm going to talk on what are the technology we are using in computational thing for computational chemistry for our drug discovery process, followed by some of the research paper what we are published with as an example for that. To order that, it is an example I'm going to give, and it's a nice introduction given to me, and also I hope a variety of students, all chemistry ma'am, all are chemistry ma'am, any different discipline? Mostly chemistry, you know, ma'am. Ma'am, most of the students are chemistry only, ma'am. Chemistry.
Proceed, sir. Yes, sir.
Yeah. Okay.
Yes,
next
sir.
slide.
Yes, sir. All of them are chemistry students, sir.
Okay. Next slide, sir. So in this bioinformatics or computational biology, what we are doing, it is a combination of subject. It is a required for mathematics, physics, chemistry, computer science. All together, we have to work to find a new drug. Among this computational chemistry, role of the chemistry is very important. Whatever the drug we to finding nowadays available, it is with the computational method we can minimize the time. That is a process rightly we are doing. So we predict the next slide, sir. So next slide. So in this computational chemistry, there is two methods. We can classical method as well as we can traditionally what we are following to finding a drug through the lead molecule, lead identification. Usually, whenever a drug classical method, whatever the compound is available, we are many trials. In the sense, will be thousands of trials, sometimes millions of trials they used for the finding a drug. Now we have to now the scenario has changed. From each disease, we have to find a particular target. Among the target, in the sense is most of the drug targets are proteins. Whatever the drug we are using, currently we are using for a variety of the diseases, all those drug targets are proteins. Some are nucleic acid, some may be a lipid, some may be a carbohydrate, but majority are proteins. We have to find out which part of the protein, which protein is particularly binding with the specific drugs. Then we have to find out that, and also particular each disease, some protein overexpressed or will be down-regulated, upregulation will be happen in each case. We have to find out, accordingly you have to find out a agonist or antagonist molecule against for this particular target. Thus we have to identify which protein overexpressed or will be down-regulated in each case. We have to find out, that is our target identification. Then followed by, we have to synthesize, we have to track, in the sense find out the which part of the molecule it is a binding, and also where will be have to compound we have to synthesize. After synthesizing the drug, you have to, in the sense through the target identification, that the compound and the protein have to bind together by means of docking methodology. Then followed by whether how it is a mimic like inside the system, we have to test it by means of molecular dynamics methods. We have stimulate it. Whatever the conditions is there in the human or animal system, same kind of conditions are given by artificially, it was given in the computationally. It was by dynamics method, all the pH and all the pH and other water molecule, all these and buffers, everything artificially it is given. Then followed by dynamics, the compound by how it is binding, score, how much will be score, high score we are getting it, minus in the sense will be negative score, how much we are getting it. Similarly, any fluctuation, without any fluctuation, how it is a binding, interaction, how many interactions, whether it is a following the Lipinski rule of 5. That's what we are compound ID synthesized, and followed by ADME predictions. In the sense will be absorption, disperse, metabolism, and excretion and toxicity. All together you have to check for each compound. Then followed by that checking of all these things, we have to check for the next level, whether it will be working in vitro conditions or in vivo condition, how it is working. Then finally it was delivered to the specific individuals. We have to find out that whether the drug is working or not. It's a whole process. This is what will be computational biology is doing. It will be from lead identification to the checking the optimization of the drug, and checking with the lead molecule with the in vitro and in vivo method all together. It is a computational process. This is what will be really computational chemistry playing a major role. Previously, each molecule, you have to each molecule whether it will be working for the particular disease. We one by one we keep testing. Then from that we have to find a compound will be identified. That's why it will be compounds or will be particularly medicine. Number of the medicines it is a very less when compared when there is a different strategy of treatment. In the sense where if you are taking a Siddha, allopathy, homeopathy. When allopathy is a method, we are for scientific method we are following it. Homeopathy and other methods also, they have the medicine, they have the crude extract, they will use it. Here allopathy, purified compounds where will be used as a thing. It is followed by Lipinski rule of 5 and other things. It was followed. It was used in the method. It is a scientifically proven method. But allopathy and where will be homeopathy and other things, it is used as a some of the medicines as a whole extract. It is used as a thing. So it will be multiple targets is going to bind. In allopathy, whatever drug we are giving, it is a specific target will go and bind, and also metabolism also happen, excretion also happen. It is a well-known mechanism. Whereas in other kind of methods, mechanism not clearly understand. That's why it will be allopathy. It is widely followed by the people. So computational chemistry, it was a help for the finding a new drug, as well as we can, if it is a new molecule you are identifying or you are identifying as a drug, it can be patent also. We can go one by one. We can see next slide, sir. So why these molecules are needed? Every day, new kind of new diseases also arise. For example, past 4 years back, COVID, and also variety of other bacterial infections, viral infections, AIDS, Alzheimer, bacterial infection, anthrax. Lot of new things also arise, and also whatever medicines, to finding a medicine for that also. Lot of people are working, for pharma industries are working, particularly chemists are working, because even whatever we are finding, final delivery part of the chemistry, it is done by chemistry people only. But fine, in the sense, purest quality, going to synthesize by chemistry only. That is made by the tablet, in the form of tablet also, it will be made by the chemistry people. They have the big role, but other people contribution also nowadays very important, because it has a lot of whatever drug they're synthesizing, it will low efficacy because of what happened, because multiple mutation happen in the individuals also. Each medicine, whatever finding against the diseases, is not uniformly response to all the individual because of the mutation in the target sites. It is also play a role with the efficacy, and also some of the things has a side effect also. For example, even our COVID vaccines, whatever we have taken, medicines what we have taken, all these has some small amount of side effect also. So that's why, to avoid these things, you're finding through the computational method what we are finding, you have to finally we have to check through the experimental methods too. Downstream of the things, similarly, sustain to the industrial academic. These are the new drug, in the sense new drugs are needed for the, because it has to, for example, first Disprin, there is a drug, it is used for the cardiac related problem, it was used. Now that recently Disprin is a drug will be used, it will be through the drug repurpose mechanism, through the computational chemistry, it was identified that Disprin can be used as an anti-cancer agent also. But the company, they taken the approval for the using in 20 years for the patent. Now again the company, they asked another 20 years for the cancer treatment, but the FDA, the approving agency, they refused it, because once it was given for 20 years, now will be other people also started synthesis and use it at a cheaper price, use it for the public use. So that now that Disprin drug, it is through the drug repurpose mechanism particularly, and that's what will be computational chemistry. It has a big role. Whatever the already approved drugs available in the market, it can be checked against the current drugs. That's what will be for, it was done in COVID also. In COVID, initially we have tried all the viral drug. It was tried against what approved viral drugs, rifampicin 2, all the viral drugs tested against to the COVID. And also bacterial antibiotics also used against the COVID. Now, finally they proved some of the drugs are they approved for the sense of controlling the COVID, and similarly vaccine also developed in such a way that only. So in the sense will be whatever drug we are identified, that can be by computational chemistry drug repurpose mechanism, that can be analyzed and used for the different applications. So that's why will be need of the diseases also increasing the population, increasing the variety of the diseases. So it's a need, so it's a need of the society to identifying the new molecules, new drugs for the betterment of the human life. So next slide, sir. So the chemistry, even multiple, all chemists, they're working together, also finding a new drug without, if you are doing in a classical method, finding a new drug, it takes very, very long time. That's why we have to apply the computational chemistry for the approach. Through the computational approach, we can reduce the time as well as we can use it for a better drugs. Next slide, sir. So this is how in the next here, whenever finding a drug, so two approach. One is curing the disease or managing the disease or chronic diseases. What is managing a disease? What is curing a disease? For example, bacterial infection. Any bacterial infection, antibiotic, azithromycin or kind of thing will be antibiotic, will be specific antibiotic was given. It will clear the 80% of all bacterial infections. Remaining 20%, it make a resistant variety, multi-drug resistant varieties or multi-unique variety of the bacterial things. It can be completely, we can, by bacterial infection, antibiotic, through the antibiotic, we can control it. Viral diseases, some of the viral diseases, hepatitis A, hepatitis B, those things can be, we can control, we can cure the disease by means of antibiotics. By specific medicines, we can cure it. Some of the medicines, we can manage it. For example, hypertension, diabetes, those things we can regulate the BP, we can regulate the sugar level through the metformin, or will be enofril, or will be kind of, these are the drugs can be used against the hypertensive patient to regulate the BP or regulate the sugar level, can be managed the disease. Some of the chronic diseases, there is no specific kind of treatment, but that is also management kind of thing. For L-DOPA, it is needed for the many of these Alzheimer kind of Parkinson, Alzheimer patients, it is needed. It is a L-DOPA directly it was given. So this is how will be some of the cases it has this kind of disease has a economic impact, and similarly it was research. It will be some of the identification of this computational chemistry method will be used for research purpose. So this is how will be a importance of the drug treatment to the disease. These are the different strategies we are following in a drug treatment. All these cases now computational chemist it has a big role in this case. Next slide, please, sir. So when you are looking for in the sense a drug synthesis, any drug synthesis, it will take 10 to 15 years. You may ask a question, sir, COVID-19, it was reported in the 90, within 2 years, vaccine was given to all the people. How it is possible? How it is possible? Whether it will be, that is the reason why we are facing the side effect of some of the vaccine, some of the drugs also we have a side effects, because really want to test the molecule, it will very long time it has to test it. The sense will be you have to identify the molecule, right molecule through the computational by means of bioinformatics approach. We can identify the computational method. We can identify the molecule, right molecule in a very short span of the time. We can reduce the 5 to 10 years of that. 5 years we can reduce it. But rest of the phase we will not possible to compromise. If you are compromised, definitely it has some side effect. It's going to give to the people, not to all the people. Part of the people may affected by means of these side effects. For example, once a drug was identified, it will take 5 years to 10 years. 5 to 6 years it will be, we have to take the identification of the drug. Later, preclinical trial, it is take 1.2 to 2 years it will take. And real clinical trial, phase 1, phase 2, phase 3 clinical, you have to do it. The phase 1 clinical trial, we have to use the 100 people, you have to use it. Minimal number of people, 100 people. If it is a phase 2 clinical trials, a minimal 500 volunteers should be used. In the phase 3 clinical trials, around 5,000 volunteers should be used. After the passing these cases, it will take the phase 1, phase 2, phase 3 clinical trial. It will take 5 to 6 years. These where will be we are missing these only for in our COVID cases. We are not done a clinical trial for this much time. Time was reduced. Not if a few months interval, it was directly given to the patients. Okay. Similarly, after that, post FDA and after this clinical trial was done, Food and Drug Administration of agency is there in the US. Similarly, it is a similar agency is there in India also. That should be approved. The company, that agency should be approved the drug. It will take a final approval. It will take 2 years. It will around. Then post it will be given to the patients. It will be trial. It will be post surveillance. It should be also tested. This is a time it will be take for 10 to 15 years for the synthesizing or introduced to the market and finding a drug. It will be taken 10 to 15 years. Approximately for each developing a drug will be cost about 1,000 million. In the sense will be in the some of the cases, many of the drug will be fails in the clinical trials also. So if you are finding a drug, thousands of the compounds, it will be finally 1 or 2 compounds only it will be come as a medicine. Rest will be neglected. Understand what we can use? If it is not the what we are tested, that can be used for the some other applications. Some other diseases can be tested. That's what will be nowadays the people are doing it through the screening method. All these things through the computational method. Once up to FDA approval, any teratogenic effect, mutagenic effect it is observed. Those things can be taken for the some other applications. That's what will be now we are doing it. So next slide, sir. So this is a pipeline of the thing. Discovery, it will be about 5 to 10 years. In the sense, a target. In the sense, whenever you are finding any disease, among the disease, even COVID also. Different targets, M protein, H protein, and the sense will be different regions we are identified. People are, in the sense will be from the scientist reports, which region is a more conserved and early segregating regions and more controllable regions. Those regions were selected for the as a target protein. Those only will be used as a drug target as well as vaccine target. Both it will be used, so the target will be used. Then lead should be discovered, optimization of the lead should be optimized. Then that lead should be by means of ADME property. What is the ADME property? Absorption, dispersion, and metabolism, elimination, toxicity, everything should be things. all the cases, medicinal chemistry, it is majorly involved in a optimization of the drug as well as ADME property checking and clinical trial, phase 1, phase 2 thing. Then it was taken to the market. This is how it will be total pipeline. It was working in the chemical drug discovery process. But all these cases, initial phase can be done with only by means of computational people, bioinformatics people with the knowledge of biology knowledge and chemistry, all the programming knowledge, everything they should have. Then rest of the things will be checked by the hardcore chemist people only, synthesizing the molecule and testing it will be done by chemist. Next slide, sir. So before starting our thing, if you're how this drug, synthesized drug, it is working. Where will be if you're taking a DNA, it is a nucleotide molecules. The DNA is a negative. Where will be all negative charge when our molecule, it will be negative charge molecule. When coming as a protein, amino acid, it is not a fully negative, fully positive. Some are neutral, some are positive, some are negative, some are amino acids too. So each one will be coding, non-coding regions all together. We in the sense from the DNA, from DNA transcription, translation, it will come as a protein. The protein is a functional form and also it will be used as a target for our whatever we are drug we are synthesizing, our molecule what we are synthesizing, protein we have to test it. How it was, protein should be, we can use as a target. So finally we are in the sense based on the boundaries of the protein or molecular energy of the protein. Our drug also should be, for example, negative molecule of the protein, active site of the protein will be negative charge. Then our drug should be a positive. Hydrophobic, hydrophilic nature of the boundaries should be exactly where to calculate. Then you have to screen for our drug molecule. Next slide, sir. This is how will be we have to identify the molecule. Next slide, sir. So before we are starting for our molecule, preliminary thing of the thing, it is a sequence alignment. Any of the protein was identified, the DNA was identified, and the sequence should be identified. We have to compare the sequences. How much gaps are there? How much in the sense will be in between sequence are there? Because it will be useful for the designing the. If it is a protein structure is not available, through the computational method, we can predict the structures of the protein also. That way we can synthesize that. We can introduce the even protein structure is not solved by crystallographically, NMR method or electron microscope method. Crystal in the sense will be electron microscope. Nowadays, crystal structures are solved through the sense our protein structures solved through the high energy electron microscopes. Not majority are solved by electron microscopes. That will come later. So a large set of the sequence either it can be by global alignment or local alignment. It is a large set of the sequence. If we have big molecules are there, you have to each molecule it was compared one by one. It will be wherever it's matching. It will be bars we can see. Wherever other things are not matching, it will say dash lines are there. So we can identify some regions are matching, some regions are not. In the sense there is a gap. So this is how we can. First step of the drug discovery process is a sequence alignment. You have to go for sequence alignment. Next slide, sir. So when going for a multiple sequence alignment, either for a simple it was shown here COVID virus, different COVID virus of SARS-CoV and bad COVID, all the different COVID viruses we are compared by means of multiple sequence alignment. So some regions or some boxes you can see here. So those are conserved region for all the species. All the species. Similarly, flu virus also initially it was swine flu. Only it will be affecting sometimes swine, sometimes it will affect the birds. The sense will be later developed variety. It will affecting both swine, birds as well as humans too. The sense will be horizontal, vertical transfer. Both things happen. So we can to develop a vaccine. We have to compare all the sequences. Through that, we have to treat all the species through the single vaccine, single type of vaccines or single type of medicine should be treated. That's what will be. These are the computational method will be used for finding new things. It can be finding the homology can be done, determination of the sequence origin, how it was origin, which species it was origin, phylogenetic information can be we can achieve, finding the specific sequences and the constructs of the evolutionary tree also by means of this method can be done. Next slide, please. So, and This is how it will be a top one will be comparative genomics, comparing all the sequences where that sequence derived from, and also phylogenetic analysis, and also gene duplication, variations, and RNA of that sequence, how it is responding. Each population wise, what is the variation? In COVID virus, each population differently responded. And also, and vaccine also it will be, that's why side effect it will be happen in a different population, different side effects too. And also, and if we're coming for a structural comparisons by modeling, homology model, once modeling was done, we can compare the mutant variety and wild variety, we can compare it. Similarly, therapeutical drug discovery, how it is a ligand, mutant variety, how it is responded, hierarchical things also we can compare by means of through the multiple sequence alignment. These are through the sequence alignment. These are the possibilities we can do for our computational chemistry. Go to the next slide, please. Next slide, please. So, structure of the protein will be, once we direct simple sequence structures are available, primary structures because only amino acids are there. Then it will go for the helix, loops, beta sheets, alpha helix, all the things at secondary structure. Each thing we can compare it by mutation. It can be represented by primary structure, whatever present, it will be reflected in the secondary structure also. Secondary structure variation, it will reflect in the tertiary structures also. Some are thing, it will be, for example, hemoglobin, myoglobin. All these cases are good example of quaternary structures. Domains are the 2 domains, 3 domains are there, 4 domains it will be, subunits are there, it will be primary reflect. And also, mutant varieties, it will be present in the primary structure, it will reflect up to tertiary structures also. Functional form also affected by means of things. For the next slide. So, next slide, sir. So, now if it is a structures of the protein available, we can solve the structures that can be through the crystallographically solved structure. We can PDP-ID, we can retrieve the sequences, we can use it for the drug discovery process. The structure is not available. Structure of the protein is not available. We can, by homology modeling, we can identify our targets. Molecule we can comparative modeling. By homology modeling, we can synthesize, we can model the protein structure. That's what will be target sequences with and also, please click, sir. Target sequences. Once we are adding target sequence, we are identified. We can whatever the sequence you want to predict, the target sequence we can retrieve it. It can be compared with the template sequences. Simply we can, what are the things should be. Next slide, sir. Next, please click it, sir. Then if you are comparing, what is a model template to be compared by means of blast analysis? There is a tool is there in a bioinformatics. We can compare that sequence similarity, pairwise alignment. Which sequence has highest similarity, that is taken as a model. That is, for example, the sequence was from the E. coli, it was done. So other variety of the bacterial species can be compared with the E. coli. Otherwise, rat sequences are available. That rat sequence can be compared with the human sequences, or human sequence to the rat sequences. Like this, we can compare, that particular protein, because in human, we have around 30,000 protein. Rat also, it has 28,000 proteins. And other higher organism, model organism, what we are using also, similar to the human, it has around 28,000 genes are available. So whatever the genes available with the human, similar kind of mimic genes also present in other organism, higher organism, mammals also, that can be used as a model. So that's what we are, first we are taken the template, then template selection. Template was compared with the target sequence, compared with the target template sequence. Then after the comparing, through the homology modeling, go to the next click it, sir. Please click. So homology modeling, we can Swiss model, or will be tripos, or different homology modeling tools are freely available. It will compare the both the sequence, given sequence as well as a template. We compare it, it will model and give it us. This is a common, in the sense we are doing it modeling by ourselves with we use with the help of some tools. Next slide, sir. Next slide. So in this, minimize the, then after the model was done, we have to minimize the model. Then minimization of thing, it was checked through the Ramachandran plots. Whether model, what we are modeled, it is a model is a correct, it will be a disallowed region. Because you all chemistry, you are studied Ramachandran model, right? So what are the, how many amino acid falling in disallowed regions? How many are in the allowed regions? Then through the ProCheck, ERET. These are the different tools used to the what we are modeled is correct or not can be checked through the different models. Different software tools are available. Through this we can check, then that model can be taken for the drug discovery. Next slide, sir. So through the homology, once we are done the homology modeling, through the database search we have to compare it, and also we are modeling whatever the through the model building we are framework it was done, and then we add some more the molecule whatever needed, the hydrogen bonds, loops, side chains needed, that was through the comparative model we can add those to the that protein. Finally model was optimized. That model was evaluated by means of this dynamics. We check the model, we optimize the model. It can be the model was taken for the further process. Next slide, sir. So once it was model was done, it was directly taken for the two method, computational method. One is a structure based method, another is a ligand based method. But in the structure based method, protein molecule by means of it is a the sets of the proteins are available. Druggability predictions are docking based on the virtual screening that the drug if you are through the different millions of compounds, millions of ligands are stored in the different databases. Many chemical databases are freely available. The databases, what are the protein we are minimized protein, against that protein compound will be allowed to bind. Each compound, millions of compound, it will be come and bind to the each this protein. One by one it will come and bind. Then whichever the molecule giving the highest score, negative score, highest negative scores giving, that molecule will take it as a top 10 molecule or 100 molecule, will take it as a for the further testing, further filtration we can take it. So like in the sense will be already drugs are used. Yeah, for example, some disease it will be against some disease, diabetes, metformin is used as a drug. Take as a metformin as a drug. What are the in the metformin to bind to the which protein it was binding? Already it's known that which protein, some of the diabetic protein, more than 150 diabetic protein are available. Among that, where it is metformin also it is known that which part of the gene it is binding or which part of the protein is binding. It's known that which what are the interactive sites. The sense will be whether it will be find the Lipinski rule of 5, hydrophobic, in the sense be donors, acceptor, molecular size of the thing, everything it was standardized. That molecule, what is the hydrophobicity, everything we have to check it. Then based on that in the sense pharmacophoric features, we have to find what are the molecule which has this kind of property in the database. From the database, that molecule all we collect together, then it was bind against the this known protein. Then we can get a new, apart from the metformin, better than metformin, other molecule, whatever available compounds are available from the databases, it will report to us. That can be further tested in vitro, in vivo method and phase 1, phase 2 clinical trials. It will be developed as a new drug molecule. That's why we are developing either structure-based molecule or ligand-based thing. We have to finding a new drug, lead molecule will be identified. That lead molecule further what taking for the testing. This is a general procedure we are using for the computational method. We are using for the finding a new drug molecules. Next slide, please, sir. So this is a methodology we are following. A drug, whatever the drug it was, once we are taking from the mouth to rectum, up to it has to pass different variety, in the sense variation with the pH, temperature, everything it has a different stage. We have to pass through the different thing. So in the mouth it is a pH of 6.5. It will reach to the alimentary canal. It will be it will reach the 2. Hydrochloric acid is there. Then rectum it will be up to 10. It will reach it. So whenever the drug is taken swallowed by means of water, it will be dissolved. Small molecule through the chorionic villi, everything will absorb. It will reach the bloodstream, and also in the plasma it was circulating. Drugs, whatever the molecule we will be taking, it was dissolved in a small fine particle. Each one it will be molecular size around less than 500 Daltons. Then only it will be a druggable molecule. Whatever we are taking the drug, all the drugs are less than 500 Daltons. It is one of the rule. It is a rule. It is called as a Lipinski rule of 5. Number of interactions should be less than 5. Number of acceptor donor, it should be in 10 to 5. Similarly, size of the molecule less than 500. So there's a log p value, everything it will be some of the limited should be limited. Then only it will be circulated to the system directly to the capillary. In the sense simple diffusion, it will reach to the target. Otherwise, some of the energy is needed to the deliver to the target molecule. So once the drug was taken, the free drugs in the plasma it was circulating. Then it is east to the right. It will be plasma binding protein. It will bind. Similarly, after through this, it will reach the target receptor. It will reach. Then pharmacologically, it will be bind to the therapeutical responses. It will do. Once the responses are over, then it was degraded. It will reach to the relieved as a through the fecal matters or through the urine. It was relieved. Some of the cases, no pharmacological responses. Therefore, the therapeutical actions needed. So it will be how it was. If it is, that is also eliminated. Sometimes it will be toxic to the system. This will be once the drug was taken, it was taken to the system. So in the sense through the plasma, it was circulating, and plasma binding protein will some of the molecule will bind to the receptor. It will reach the receptor. It will do their functions. This how will be things are by it will be mechanism will be binding in the drug molecule. Next slide, sir. So major part of our drug binding is a blood compound, plasma protein, and blood cells. Whatever the specific blood cells in the sense it's a specific proteins. Extra tissues also protein, fat, bones also will be binding to the some of the drug molecule binds to this way also will be binding. Next slide, sir. So whenever we're going to the thing, some of the molecule it will has action. It is sorry, sir, it was disconnected. So in this binding of the next slide, sir. Yes. So binding of the drug to the human serum in the albumin, it is also will be differ from different side. For example, warfarin, diazepam, tamoxifen. These are the different drugs in the protein. The binding site also differs. Each molecule, warfarin is the size of the warfarin is different, diazepam is different, tamoxifen also differ. So it depends upon the size, it will be binding also changes. Go to the next slide, sir. So this how will be through the polymer chemistry, how will be, what are the things are needed for here? All the knowledge, genetic knowledge of the genetic knowledge, what is a mutation, what is a protein information, similarly synthetic chemistry, biology, environmental nature, polymer nature, everything needed for the all together will be needed for the synthesizing a molecule. Without, if you are ignoring any of these, also it will be toxic. Because I'll show you the example. Go to the next slide, sir. So this how will be a target identification, target validation, lead identification, and a lead optimization, preclinical trials. In all these cases, if your bioinformatics approach, we will be it has a role in the target and validation. It will be used in the bioinformatics. Structural biology, it has a big role in the target validation, lead identification. Cheminformatics, it has a lead optimization, lead identification, and preclinical trial, all together it will need the cheminformatics, social biology is needed. So all together it will be as a the sense will be needed for the synthesizing the drug. Go to the next slide, sir. This is how will be a what are the things needed for the drug discovery, literature document, literature, and also high throughput screening. What are the how drug was identified through the high throughput screening? It will millions of compound within a day it was screened, and also we identified the lead molecules. Then through the computational identification chemistry, lead was optimized, and then ADME property was checked, and data through the preclinical. These are the different substeps are used in the things for our drug identification. Drug identification. Go to the next slide, sir. So these are the different chemical databases. Once people are working in some plant as well as animal species, who are working in a different thing. Once they identify, their compounds can be deposited in a chemical data banks. We are some of the compound we are retrieved from some plant compounds, and also jamun fruit as well as a different compound we are identified. All the things were deposited in the chem bank. Why we have to deposit? Whenever we are depositing, that can be tested against to the target, as well as other people who are working with the same kind of work. Also, they can retrieve the compound from those databases. Because why I'm telling here, we identify a few compounds. It is from the jamun fruit, as well as we are identifying from there is a rubox. There is a species from the plant species. The same compound present in aloe vera also. That compound we are used against the anti-snake venom. We are used it. That compound is present in aloe vera also. It is the same compound is present. So we find out. So instead of the same effect can be taken from aloe vera also. Okay, that's what will be zinc databases, PubChem, binding databases. These are the different chemical databases available for our study. Next slide, sir. And also next slide. So these are the next slide, sir. This is how it will be a different drugs where will be what are the content present. For example, drug database, it has a detailed drug data, comprehensive drug targets, are what are the drug banks are available, what is the role of the drugs, and also everything description it was given here. So it will be, for example, bioactivity in the sense inhibition activity or dissociation activity, half max, half minimal. All those things can be bioavailable. Each one it will be stored in our data banks. We use this as a reference models. We can compare whatever the data we are working on it. It can be compared with these kind of databases. Go to the next slide, sir. Next slide. So how PubChem work? So we can go to next slide. Next slide. Next slide, sir. So this will be sometime by PubChem substances are available. What we have deposited in the data bank, compounds are available. Assays also reported, and also compounds are available. Substances are available. All the information we can put together, because whenever we are working with a compound, that assay information also we have to take it from the PubChem data banks. These are the things we can use it for the. This is how will be computational things we can retrieve from all the cases. Next slide, sir. Okay. So in this, more than 10 online chemical databases are available. ChemDraw, Marvin Sketch, these are the different tools used to draw the other chemical structures. Whatever based on that available structures, we can draw our chemical structure. We can upload to our other chemical, further downstream process. Next slide, sir. Next slide. So in this, single nucleotide polymorphism has a big role, particularly on proteins. How the DNA to the protein, for example, altering of the DNA nucleotide sequences, protein level conformation of the protein also changing. It will going to influence on our drug binding also. It is going to influence with the drug binding also. We have to accordingly you have to check. These are nowadays people are concentrating on the where that mutations also involving our drug discovery process. Next slide, sir. Next slide. So different type of mutations are reported. Maybe nonsense mutation, missense mutation, synonymous, non-synonymous mutations, coding regions, non-coding regions, intronic, exonic, promoter variations, 3' variations. These are the different variation in the genes. It going to influence the protein structure. That protein structure, it is a target of our drug target. So changing in the drug target, efficacy of the protein are also going to change. This will be playing a major role. Changing in the protein, changing it will be going to influence on the responses of the chemical level also. Next slide, sir. Next slide. So people are for people, same drug, 4 different people differently respond. Some people may not respond. For example, Calpol we are taking. Calpol, maybe some people need the 500 mg, some people need the 600 or 650 mg, some people need the 1200 mg, some people need the 125 mg. Sometimes some people are allergic for the drugs also. So we have to, because of the mutation in the drug targets, these wide variety of changes also happen. So we have to, when it's synthesizing a drug, you have to concentrate on our mutation part also. Next slide, sir. So diversity people, for example, human diversity, it has a big role. So optimal responder, some people are suboptimal responder, non-responder, adverse reactions also. This will be a group of people from the same drug. Some people differently responded, and also based on the, for example, based on the people group 1, drug A will work, B some other drugs work, C some other drug will work, some other, in the sense another drug standard treatment, unusual genetic profile, so it may not be work, and adverse reactions also happen with some people. So whatever drug we are chemically we are synthesizing, it should be responded different to the people. So we have to, accordingly we have to find what are the mutation associated with this. Also we have to find out, accordingly we have to give the treatment. To the next slide, sir. Next slide. So genotype 1, it is a where will be responded in, for example, this mutation normally respond. Regulatory side, all these particular PRP alpha protein will be co-activated and other proteins binding. So there is a regulatory site changes. So genotype, these mechanism of multiple protein not binding properly, what happen? Gene expression may not be properly happen. The genotype 1 responders, genotype 2 non-responder for the medicine. Those people should be avoided for the particular treatments. Go to the next slide, sir. So this is how will be once the drug was taken through the oral, if you are taking absorption, distribution, metabolism, all the things happen in a different part of the system. So everything is a pharmacokinetic process. How it was responded, if it is a even with any mutation in the pharmacokinetic way going to affect it. Mutation in the drug target as well as expression with essentially metabolic pathways, mutations affecting the pharmacokinetic process. Go to the next slide, sir. In pharmacodynamic process, once how drugs are responding because of the mutation happened in the individual, ion channel, whether how it is ion channel will be working, how it is working in a transporter mechanism, how it is working in an enzymatic mechanism, how it will be the receptor mechanism, how it was responding. So it depends upon the dynamic drug molecule, how responded in the system will be playing a role with by means of dynamic process. This will be a variation in the ion channel, enzymatic mechanism, transporter, receptor, all these things influence the mechanism of pharmacodynamic process of the drug molecule. Next slide, sir. So protein molecule, next slide, sir. Next slide. Next slide. Next slide, sir. It's a movie, it's not playing. Go to the next slide, sir. So, okay. A good example, thalidomide, there is a drug, well known drug, not properly. What happened here? Particularly the drug used for the in the sense to avoid the pregnant women, particularly on the 1940 and 42 time it was given. Company, whole company was closed. Go to the next slide, sir. Next slide. So what is the response? What is the response for the here? There is a methyl group present in the this molecule. This methyl group. There is a methyl group present in this NH2. So because of this present, it is giving a teratogenic effect. So what happened in the sense there is a carboxy group. It is instead of methyl group, it was added a carboxy group. Once this carboxy group was added, then the teratogenic effect was reduced. Now by means of drug repurposed mechanism, the drug was used for the leprosy patients. Instead of that, previously the drug was used against the teratogenic effect. It was reported with the teratogenic. It was banned. The drug was completely withdrawn. The company was also closed. Through the drug repurposed mechanism, the drug was again identified. It was used for the by means of leprosy patient was used. This is how will be still people are working to reduce the teratogenic effect with that by modification with the reduce the high cost of the things also reduced by means of computational method. We can reduce it, and also toxicity also reduce applications. Initially it was reported for the in the sense of vomit and nausea. Now it was used for the anti. So these can be further. Next slide, sir. Pharmacophore. Next slide, sir. Next slide. So this all be a pharmacophore models and features can be used. Like in, for example, pharmacophore in the sense will be what are the pharma in the sense acceptor, donor, hydrophobic, hydrophilic regions. All these features will be put together. Then we have to screen this through the virtual screening. Then we will find a right molecule. We can pick it up for our study. Go to the next slide, sir. In this, we have to test set and trial set. We have to. These are the features: hydrogen bond donor, hydrogen acceptor, anionic, cationic, hydrophobic, aromatic things. All together in the what are the regions present in the protein? Accordingly, we can features we can set that features we can screen against to the databases. That molecule, what are the molecule is available with that features, it will bind to that protein. Then that molecule can be synthesized, tested against the particular target molecule. This how will be computational chemistry. It will work for the developing a new molecule. Go to the next slide, sir. Next slide. So, for example, this how will be a acceptor, donor, hydrophobic, donor, acceptor. This is a feature. It is present in the protein. So we are designing a hypothesis: A, D, H, D, G, two donors and one acceptor, two acceptor, one hydrophobic. 5-point pharmacophore. With this score, if you are screening, what are the point of these kind of features available in the per ligand molecule? That molecule will bind to this protein. So finally, one molecule was reported with this. So that molecule can be taken for the synthesis. It can be used against that protein molecule. Go to the next slide, sir. Next slide. Next slide, sir. Next slide, sir. So next slide, sir. Next slide, sir. Sir, next slide. So next slide, sir. Okay. Okay. Okay, fine, sir, fine. So key is quantitative structure activity relationship. It is a thing. It will be used for our. This is a. Previous slide, sir, previous slide. Previous slide, sir, previous. Okay. These all will be a pharmacophagers. It is one of the research work what we have done it. So pharmacophagers, it is there in some of the active site, and where will be acceptor, donor, all the things we put together. We screen against the databases, and from this we can set our IC50 value. We set accordingly. Quantitative such activity relationship also test set among the compound. We are whatever the mutation will record, we modify that mutations. Then from that will be the database will screen. Some of the molecule we will keep it as a training set. Some are used as a the test set molecules. That those things will be minimal 20-30% of the molecule will be a test sets. Remaining will be a training set. Accordingly, we can identify the molecule. That will be taken for the synthesis part. That can be taken for the drug molecule also. Go to the next slide, sir. Next slide. Next slide, sir. Next slide. Next slide. Whatever this region, it will be the most active compounds and least active compounds. Most active will be in blue in color. It is a least active compound. It will be more red in color. These were where will be our inside that ligand molecule. Protein will be where will be protein ligand how interacting. It was shown in the map. Most active compounds. It is a binding in a blue color. Least active compound will be showing in the. It will be in a orange or it will be red color. We can compare. We can select your molecules. Next slide, sir. Next slide. Next slide, sir. Next, sir. Yes. Now the computational chemistry, what we are still now we are done. That is where will be pharmacophore, QSR model. What we are done, synthesizing, homology modeling, everything. Now the whole total scenario get changed because of artificial intelligence and neural network. Large chemical space. It will be generative modeling. It was done by means of algorithms were written. Many of all the things through the future perspective of this computational chemistry based on this model only. Large chemical space it was by algorithm. Then we generate a different model. Go for the case study, sir. In this case study, there is a we want to say this. This is our one of the example. Snake venom will be whenever snake venom. Next slide. Snakes are very probably in thermal where will be India kind of places, India, Pakistan kind of Southeast Asia. In the sense only poisonous snakes are very high, and these snakes, particularly two variety of major snakes are available, Naja and Crate variety of the snakes. Each type of snakes it has a different poisons for producing mechanism of killing the people in a different mechanism. One it will be used as a SVMP protein is released. Another one case phospholipase kinase protein was released. So in our study case, we identified phospholipase and also there will be zinc ions. In the sense, what will be it will be playing a major role. We analyzed through this molecular dynamic simulation method. We identified by means of one of the metal or SVMP protein through this protein we are analyzed. Sir, slide is not possible to see, sir. Sir, slide not possible to see, sir. Slide we are not possible to see, ma'am.
Maybe I think that studio time is over. 1 hour.
Okay. Okay. Okay, ma'am. Okay, okay. That slides are available already shared, ma'am. Please give it to the people also. Whatever the study based on what I am given in the talk, the whole thing it was by example. Whatever the this kind of computational chemistry, what we had done in our thing, it was example as an example. It was given in the PPT. Please, from ma'am, please collect the PPTs. See that examples. Some of our research publications based on the computational chemistry method, how we are develop chemistry in the sense will be drugs for the using the computational chemistry for the snake venom, because snake venom it is only vaccines are available. From our case, we identified a chemical compound from a plant. That plant compound it can be able to control both the SVMP. We can inhibit the SVMP as well as PLA protein. And also both variety of the snakes also. That compounds able to inhibit both expressions. It can be instead of using for the initially we are going for a doctors. They will for snake who are the people who are snake antivenom in the sense bited. Doctor will ask which type of snake was bited individuals according to the vaccines were given. Now or will be polyvinyl in the sense will be there's antivenom whatever they're giving. Multivenated things was given. Now, through these drugs, they taken these particular drugs, it will be save the people. Whatever the drug, whatever the type of the snakes will be bited, in the sense will be snakes was bited. This, if you're giving this drug molecule, both type of the people maybe type of people who are bited by the snakes, both type of snakes also will be prevented from this kind of medicine can be prevent our things. This what will be our conclusion from this. So computational chemistry has a big role in our bioinformatics part. So without synthesizing, testing the molecule, further drugs may not be developed. Whatever we are developing, we are using, it is most of the thing as a computational chemistry models only. With the help of chemistry people only, we are synthesizing the chemicals. Trial and error, whatever the drug in the sense will be a clinical trial and all the things we are doing it. So with this, I'm concluding my thing. So PPT is also is not there. So I'm concluding here. Please, if you have any doubts, please ask us. Please, it is you can please. Ma'am, maybe session may be given for discussion, ma'am, because the PPT it is not there.
Yes, sir. Some studio time is over, that's why maybe. So someone asking, what is docking? Docking means in silico study, no? Computational biology.
Yes.
Hello.
Docking is a process.
Yes, docking is in silico study, no? Computational biology.
Yeah, in silico method only I am talking, ma'am, because I am a bioinformatics. I am.
Yes, sir. Yes, sir. Hello, sir.
Which protein is responsible for each disease? From the disease, that section of the protein we solve it. The solved section, if it is yeah. Then we will write a drug model. Yes, ma'am.
It's giving an equal answer to the in vitro and in vivo studies, sir.
Yeah, that is also same. If it is a binding, it is called as a docking only, ma'am.
Yes, sir.
If it is a binding, but a computational method, what we are telling, the same thing will be applicable for your in vitro model also. The docking, the sense will be binding. We can say in vitro, we say binding.
Without the preparation of chemicals, it is possible to possible to study or all it is useful to study the biological system, sir.
Yes, ma'am. But that's what we are doing. By computationally, we are synthesizing our compounds by whatever from the different data banks, arsenic, zinc, lot of chemical banks are available. From that, we are screening that because what are the chemical compounds freely deposited in multiple things. So from that will be which molecule can be suited. It will be by either agonist way, antagonist method. It will binding. That we check it first.
Yes,
Then
yes, sir. Thank you,
once
sir.
we are binding, we'll come to you. Chemist people will come. Sir, please, will it possible to synthesize the chemicals?
Okay,
Then,
sir.
if you are
Okay, sir.
after synthesizing by the chemist only, we can proceed, ma'am.
Yes, sir. Yes, sir. Thank you, sir. Thank you,
Yes,
sir.
that is the procedure.
Yeah,
Without
yeah, that's,
chemistry, the further downstream process of the things not possible. We will not possible to make it as a medicine.
yes, sir. Yes, sir. Yes, sir. It's very important in a pharmacological study, no, sir. That's why.
Yes, yes. We are working, our theme, we are working in a drug discovery process. So, computational part, we are finding drugs through the databases. Even some plant also we are working that we isolate the compound, we deposit the same compound, chem bank will deposit, ma'am. Whatever we are working, compound, we are isolated compounds, we are depositing in the chem bank.
Yes, sir. Yes. So, thank
Some
you,
of
sir.
our
Thank you.
compound also will be there in the chem banks.
Yes, sir. Thank you, sir. Thank you. Enough, sir. Enough. Thank you, sir. Sir,
Thank you. Because
I hand
it
it
is
over.
online, probably it is
Ah,
maybe physical mode, probably little more things can be given. So people also
yeah.
will interact more. Yes.
Yes, sir. Yes, sir. Yes, sir. Yeah, students
I
also very
hope
much
you all,
interested to listen, sir.
no,
Yes,
no,
sir.
yes, ma'am. Thank you, ma'am. I hope you all understand.
Yes, sir.
Yeah, it'll be, I don't know, because it'll be through the student, because we see the students also, how much
Yeah.
will be they are benefited, we can come to know that. Okay.
Yes, sir.
But
Yes,
next,
sir.
whenever next opportunities are coming, if possible, we can physical mode as well as it will be in online mode also. Some other
Yes, sir.
kind of more hardcore chemistry we can talk, ma'am. Chemistry with how
Yes,
we
sir.
are applying and other thing we can discuss.
Yes, sir. Thank you very much, sir. I hand it over the session to the vote of thanks, sir. Thank you, sir.
Thank you
Thank
so
you,
much,
sir.
ma'am.
Yeah.
Thank you.
Today topic is currently important for the all the field. Very useful explanation about the drug designing, computational chemistry, in silico studies of drug discovery and metabolic activity and biological activities, drug binding to our body. Everything, every mechanism is very useful, sir. My chemistry students are very much interested to listen everything, sir. Thank you, sir. Thank you very much. It is an honor to be asked to extend a vote of gratitude on occasion on behalf of chemistry department
Yeah.
to participate
Thank you, ma'am. Please,
international.
ma'am, please, who are interested, please share my PPT, ma'am. Nothing wrong.
Compulsory, sir. Compulsory, sir. I do it, sir.
Yeah, please
Yes,
give
sir.
it to them. And also, I
Hmm.
will share my email ID. Also, please share with them. Who are
Okay,
interested, please
sir.
ask them to contact us. We'll help you, ma'am.
Compulsory, sir.
Yeah,
Thank you,
thank
sir.
you.
Thank you very much, sir.
Thank you.
Sir. Thanks to all the participants to participate the international seminar 1 week virtual mode emerging technology in chemistry. I would also like to express my sincere thanks to our chief guest Dr.M.KARTHIKEYAN for sharing your talents and importance to work docking studies together. You have providing us valuable information, recording, sir. Thank you, sir. Thank you very much, sir. With great pleasures, I would want to express my gratitude to our management for providing this opportunity, for agreeing to hosting this webinars in connection with the international seminar Emerging Trend in Chemistry. My special thank to our principal, none other than our esteemed principal, Dr. N. Sujata, as driving force behind the encouragement and a strong belief to teamwork, sir. My special thanks to Mr. Balaji, sir, complete help of the online program, sir, for last 7 days, sir. Thank you very much, sir. I consequently owe to my department faculties and the entire teams, the participants, and my lovely students to express my gratitude. I want thanks to everyone who is yes once more. Thank you, sir. Thank you very much, sir. Your valuable subject, sir. Thank you, sir. I share
Thank you,
all
ma'am.
the
Thank
mail
you.
and PPT to the students, sir. Thank you, sir.
Thank you, ma'am. Thank you so much.
Thank you very much, sir. I wind up
Thank you.
the session. Yes,
Thank
thank you,
you.
sir.